Your anxiety is not a personality flaw. Your joint pain is not bad luck. Your chronic brain fog is not laziness. Your gut problems are not a mystery your doctor needs to solve with a prescription. They are software crashes. Your body is running $200,000 worth of prehistoric survival hardware, forged over hundreds of thousands of years in the resource-scarce, thermally volatile, biomechanically chaotic environments of the Pleistocene. And you are attempting to run that ancient software in climate-controlled rooms, on flat pavement, in chairs, staring at glass rectangles, eating every 3 hours on a schedule that would have made no biological sense to any human who ever lived before 1800. Every single chronic condition that now dominates modern medicine, the anxiety epidemic, the rampant joint degeneration, the metabolic collapse, the neurodegeneration, has the same root diagnosis: evolutionary mismatch. The biological hardware and the modern environment are fundamentally incompatible, and the incompatibility is getting worse every generation. Today we restore 7 factory settings, 7 ancestral biological programs that your body was built to run, that modern life has systematically switched off, and that you can begin reactivating today with nothing more than the knowledge of what they are. Hack 1. Panoramic vision. The nervous system factory reset. Right now, wherever you are reading or watching this, your eyes are performing a very specific, very damaging act. They are pointed forward, locked onto a narrow, flat, two-dimensional surface. This is called foveal staring. Foveal vision uses the cone photoreceptors packed into the macula, the central high-density zone of the retina, to produce a crisp, high-acuity image of whatever you are directly looking at. And your brain treats it as a threat. Here is the mechanism. Foveal input is processed through a pathway that runs directly to the superior colliculus in the midbrain, which maintains tight structural connections with the pulvinar and the amygdala. These are your threat detection centers. In the ancestral environment, narrowing your visual field to a single focal point meant one thing. Something required your acute, urgent attention: a predator, a combat situation, a life or death moment. Your nervous system learned over hundreds of thousands of years to treat foveal staring as a biological alert signal. Every time your visual focus narrows, your sympathetic nervous system activates, adrenaline rises, pupils dilate, and your body prepares for a fight. You're doing this for 8 to 12 hours every single day while sitting completely still in an office chair. Your biology has no framework for that scenario. It interprets your screen time as a permanent and unresolved emergency. And it runs the stress cascade accordingly. The antidote is ancient. Peripheral vision. The rod-dominant photoreceptors that dominate the outer visual field detect motion, depth, and spatial orientation across a much wider sensory arc. When peripheral vision engages, and particularly when you are moving forward through space creating what biologists call optic flow, the visual signal shifts away from the amygdala threat pathway. Instead, it activates the Edinger-Westphal nucleus, the preganglionic cluster of cholinergic neurons in the brainstem that drive the parasympathetic ciliary ganglion. The pupils constrict, the ocular muscles relax, cortisol drops, vagal tone rises, the nervous system receives the all-clear signal. The modern mismatch removes optic flow entirely. You sit still and stare. The brain never gets the signal that the emergency is over. The practical step. Right now, look up from whatever screen you are using. Without turning your head, expand your attention to the outer edges of your visual field. Hold it for 30 seconds. Let your eyes go soft. Notice the periphery without focusing on it. You just manually switched your autonomic nervous system from sympathetic to parasympathetic. Do this 3 times a day, minimum. Walk outside without your phone and let optic flow do the work. Hack 2. Segmented sleep. The Midnight Productivity Protocol. Forget everything you were told about 8 hours of uninterrupted sleep being the natural human baseline. It is not. It is an industrial-era invention, and the medical community has been pathologizing its absence for over a century. Historical and anthropological evidence spanning more than 500 references, from Homer's Odyssey to documented pre-industrial sleep diaries to modern tribal populations, confirms that the universal pre-industrial human sleep pattern was biphasic. A first sleep of roughly 4 hours. A natural waking period of 1 to 2 hours in the middle of the night. Then a second sleep until dawn. This was not insomnia, this was the operating system. The neurobiology of the midnight waking window is extraordinarily specific. Brainwave activity during this period shifts predominantly into the theta frequency range between 3 and 8 Hz. Theta is the same frequency associated with deep meditative states, memory consolidation, and emotional processing. Simultaneously, the anterior pituitary dramatically surges prolactin output. This nocturnal prolactin surge is deeply linked to enhanced NREM sleep, the suppression of the hypothalamic pituitary adrenal axis, and a state of profound stillness that is physiologically distinct from anything you can achieve through deliberate relaxation during daylight. Progesterone then begins its own rise during this window, organically initiating the biological signal to return to sleep. GABA and adenosine inhibit the wake-promoting orexin neurons in the lateral hypothalamus, and the second sleep begins. This is an exquisitely orchestrated neuroendocrine program, and modern life dismantles it in one move. You wake at 2 a.m., which your biology designed as a theta wave meditative window, and you panic. You check your phone. The blue light hits your retinal melanopsin receptors and immediately suppresses melatonin and prolactin while spiking cortisol and epinephrine. The meditative window becomes an anxiety spiral. The biological program is not broken. The response to the program is what breaks it. The practical step. If you wake in the night, do not look at any light source. Remain in complete darkness. Breathe slowly. Your brain is in a naturally theta-dominant state. It is giving you a free meditation session. Use it. Lie still. Let the prolactin do its work. And within 30 to 60 minutes, progesterone will carry you back under. The waking is not the problem. Your response to the waking is the problem. Hack 3. The deep squat. The biomechanical factory reset. The most damaging piece of furniture ever invented is the chair. Not because it is uncomfortable, because it is too comfortable. And your pelvis has paid the price. For the entirety of hominin evolution, the default rest posture was the deep squat, not a 90-degree chair squat. A full ass-to-grass, 110 to 135 degrees of knee flexion, thigh below parallel. Indigenous and pre-industrial populations still perform this movement effortlessly across their entire lifespans. It is not a skill, it is a biological baseline. When you squat this deeply, the gluteus maximus activates at 13 to 61% higher maximum voluntary isometric contraction than it achieves in a narrow stance. The entire trunk musculature engages to stabilize the torso intrinsically. The fascial planes of the lower body are subjected to dynamic shear, which stimulates the fasciocytes embedded in the connective tissue matrix to synthesize hyaluronin in a depolymerized high molecular weight form. High molecular weight hyaluronin is the body's own biological lubricant, binding to CD44 receptors and maintaining the frictionless sliding of muscles, joints, and fascial planes. When you sit in a chair for 10 to 15 hours, none of this happens. The hyaluronin fails to recycle. Its molecules superaggregate into tangled, viscous adhesive chains. The fascial layers stop sliding and start sticking. Clinically, this state is called fascial densification. It is literally your connective tissue turning into glue. It distorts force transmission, creates myofascial trigger points, and drives the nociceptors that generate your chronic lower back pain. And the chair does something else almost nobody talks about. It compresses your gluteal musculature passively for hours at a time, inducing what neuromuscular clinicians call gluteal amnesia. The neural drive to the gluteus maximus is progressively down-regulated. The muscles forget how to fire. When they stop firing correctly, the entire force-coupling relationship of the pelvis collapses, and every joint above and below it compensates for the instability. There is also a direct gut consequence. The deep squat straightens the anorectal angle by fully relaxing the puborectalis muscle, the muscle that normally wraps around the rectum to maintain continence. A straight anorectal angle means complete, effortless colonic evacuation without the Valsalva maneuver. The squat also mechanically compresses the right thigh against the cecum while applying pressure that temporarily closes the ileocecal valve. This prevents fecal material from refluxing backward from the colon into the sterile environment of the small intestine. Modern pedestal toilet sitting does none of this. The ileocecal valve is left unprotected, and the Valsalva pressure required to evacuate past a still kinked puborectalis is directly linked to the pathogenesis of diverticulitis, appendicitis, and chronic constipation. The practical step. Begin adding 5 to 10 minutes of a supported deep squat to your daily floor time. Use a door frame, a pole, or a wall for balance as you develop hip and ankle mobility. If you cannot get all the way down initially, put a folded towel under your heels. The target is full depth. Your tissues will adapt within weeks. If you have a toilet, consider a squat platform. This is not optional furniture, it is biomechanical maintenance. Hack 4. Micro cold spikes. The metabolic and neural upgrade. Your ancestors were cold. Not occasionally, constantly. Prior to synthetic textiles, insulated structures, and central heating, the human body was subjected to dramatic daily and seasonal temperature fluctuations. And it built an extraordinary set of survival systems in response. When you submerge in cold water, the sympathetic nervous system produces a biochemical surge that has no pharmaceutical equivalent. Norepinephrine increases by 530%. Dopamine increases by 250%. These are not modest bumps. These are systemic floods of neurochemicals that produce states of acute arousal, sustained cognitive focus, and significant systemic analgesia. They simultaneously downregulate the markers of chronic autoimmune inflammation. Metabolically, cold exposure activates brown adipose tissue. BAT is not ordinary fat. It is mitochondria-dense, heavily vascularized tissue located in the posterior neck, clavicular regions, and around the spine. Inside the inner mitochondrial membrane of every BAT cell lives uncoupling protein 1, UCP1, designated SLC25A7 in the genome. Under cold stimulation, UCP1 physically decouples the mitochondrial electron transport chain from ATP synthesis, allowing the proton gradient that would normally drive energy production to dissipate entirely as heat instead. The result is non-shivering thermogenesis, a metabolic furnace that burns glucose and free fatty acids directly without producing ATP. This mechanism is one of the most powerful tools for metabolic flexibility and insulin sensitivity available to the human body, and it atrophies completely in thermoneutral modern environments. At the neurological level, cold shock does something even more remarkable. Temperature-sensitive TREC1 channels in the brain detect the drop in temperature and rapidly upregulate a cold shock protein called RBM3, RNA binding motif protein 3, RBM3 orchestrates structural synaptic plasticity, rebuilds degraded synaptic connections, and rescues neurons from programmed cell death. It does this through a non-canonical activation of the TRKB receptor, which drives phospholipase C-gamma-1 and phospho-CREB signaling while simultaneously suppressing the destructive ERK pathway via DUSP6. In animal models of Alzheimer's disease and prion-related neurodegeneration, the upregulation of RBM3 through cold exposure has halted neuronal loss, rescued cognitive function, and dramatically extended survival. You have a neuroprotective cold shock protein in your brain that can only be activated by cold, and modern life never activates it. The practical step: end every shower with 30 seconds of the coldest water your tap can produce. You do not need a cryotherapy chamber. You do not need an ice bath. 30 seconds of cold at the end of your morning shower begins restoring BAT activity, begins reactivating UCP1 expression, and begins triggering the RBM3 neuroprotection cascade. As tolerance builds, extend the duration and reduce the temperature. The norepinephrine and dopamine spike will outlast the shower by hours. Hack 5. Sensory tracking. The neuroplasticity maintenance protocol. There is a structure inside your skull called the hippocampus. It lives in the medial temporal lobe. It is the organ of spatial navigation, memory formation, and cognitive mapping, and GPS is slowly destroying it. The hippocampus contains two types of specialized neurons critical to its function. Place cells encode your specific location in the environment. The adjacent entorhinal cortex contains grid cells which map the geometric coordinates of the physical world. When you navigate actively without external guidance, the hippocampus fires at theta frequencies, alternating rapidly between representations of past environments and current sensory inputs to enable flexible planning and detour finding. The olfactory cortex, including the piriform cortex and the olfactory tubercle, maintains powerful direct connectivity to the hippocampus, forming olfactory place maps that integrate scent signatures with spatial coordinates. The hippocampus engages in continuous reciprocal signaling with the medial prefrontal cortex to execute spatial working memory and navigation decisions. This entire computational system is governed by a strict use it or lose it principle. Active navigation in complex, nonlinear, unpredictable environments drives adult hippocampal neurogenesis, physically growing the dentate gyrus. Remove the cognitive demand and the tissue atrophies. The London taxi driver study proved this definitively. Using voxel-based morphometry, researchers demonstrated that drivers who spent years memorizing London's complex nonlinear street network, a process called the knowledge, requiring zero GPS use, developed significantly enlarged posterior hippocampi compared to control subjects. The voxel of peak gray matter density difference corresponded exactly to the navigation-related structural expansion predicted by neurogenesis models. Conversely, civilians who rely exclusively on turn-by-turn GPS directions show measurable shrinkage of this exact cortical volume. The hippocampus and the medial prefrontal cortex are the earliest regions affected by Alzheimer's disease. By outsourcing navigation to external devices, modern humans are engineering the atrophy of the precise tissues most vulnerable to neurodegenerative decline. The practical step: once a week, navigate somewhere without GPS. Drive or walk a route from memory. When you arrive somewhere new, intentionally study the route back before your phone tells you. Walk through a park and try to re-emerge at a specific exit without a map. Smell the environment deliberately. Make your hippocampus work. These are not exercises, they are maintenance. Hack 6. Biphasic eating. The gut housekeeping imperative. Your gut has a cleaning cycle, and every time you eat a snack, you reset the timer and cancel the clean. The mechanism is called the migrating motor complex, first characterized by Sershevsky in 1969. The MMC is the interdigestive housekeeper of the gastrointestinal tract. During the fasting state, the stomach and small intestine run a cyclic motility program every 90 to 230 minutes. The cycle moves through 4 phases. Phases 1 and 2 are relatively quiet, involving smooth muscle quiescence and low-level contractile activity respectively. But Phase 3 is the event that matters. Phase 3 produces a powerful, coordinated wave of intense peristaltic contractions, originating in the antrum or duodenum and sweeping distally through the entire length of the ileum over 5 to 10 minutes. This electromechanical sweep is hormonally orchestrated by cyclic releases of motilin and ghrelin, 2 structurally related peptides from the same G-protein-coupled receptor family, working in coordination with vagal tone from the gut-brain axis. The physiological function of the Phase 3 sweep is specific. It aggressively propels indigestible debris forward, and it physically sweeps excess bacterial populations from the sterile small intestine down past the ileocecal sphincter into the colon where they belong. Now here's the critical fact. The single physiological trigger that abruptly terminates the MMC and resets the entire timer back to 0 is feeding. Any caloric intake, however small, stops the cycle immediately. When you graze continuously, eating every 2 to 3 hours from waking to sleep, Phase 3 of the MMC never occurs. Not reduced. Never. Colonic bacteria are free to migrate backward into the small intestine. Over weeks and months, this produces small intestinal bacterial overgrowth, SIBO. It produces impaired gastric emptying. It produces the intestinal pseudo-obstruction and systemic gut inflammation that underlies vast amounts of modern digestive pathology. Endocrinologically, continuous feeding demands continuous insulin output from the pancreatic beta cells. This prevents the circadian insulin rhythm from ever expressing its natural trough, completely overrides peripheral glucose transporter upregulation, and drives the system toward receptor downregulation, beta cell exhaustion, and ultimately type 2 diabetes. Your ancestral biology was designed for a hunt and feast cycle, extended periods of physical activity and fasting, followed by a substantial meal. The MMC ran freely between feedings. The small bowel stayed clean. Insulin cycled properly. The practical step. Compress your eating window to 8 to 10 hours per day. Not starvation, just a feeding window. Stop eating 3 hours before sleep. Do not snack between meals. Give your MMC time to run Phase 3 at least once or twice between eating episodes. This single change, returning to a biphasic eating pattern, addresses SIBO risk, insulin sensitivity, and gut motility simultaneously. Hack 7. The foraging gait. The joint preservation protocol. Your joints are not degenerating because you exercise too much. They are degenerating because you always walk on the same surface, and your foot does not know what to do with that. The human foot contains 26 bones, 33 articulations, and over 100 muscles, tendons, and ligaments. Deep within this architecture lives a class of muscles called the intrinsic foot muscles, the IFMs. The abductor hallucis, the lumbricals, the interossae. These are not designed for power output. They are unipennate and bipennate microstabilizers, shock absorbers, and proprioceptive sensors built to process the continuous chaotic sensory input of unpredictable natural terrain and constantly recalibrate joint position, balance, and force distribution throughout the entire lower kinetic chain. When you walk on natural variable terrain, clinical biomechanical data shows what happens. Step length variability increases by 22%. Step width variability increases by 36%. Positive mechanical work at the knee increases by 28%, or roughly 0.0106 joules per kilogram. At the hip, it increases by 62%, or 0.1078 joules per kilogram. Overall, net metabolic energy expenditure rises by 28%. The IFMs fire constantly, redistributing kinetic forces across dozens of joints and fascial planes. The proprioceptive data feeding back to the somatosensory cortex is continuously variable and richly informative. When you walk on perfectly flat, paved concrete in a rigid, cushioned shoe, the IFMs receive no proprioceptive challenge and progressively atrophy. The terrain never changes. The same biomechanical vector absorbs every step. The dormant stabilizer muscles are bypassed entirely. Kinetic energy is routed directly through the primary movers and major joints in the identical pattern, repetition after repetition, with no variability to distribute the mechanical load. This is the definition of repetitive strain injury, not overuse. Monotonous, unvarying use with no adaptive stabilization. Plantar fasciitis, shin splints, chronic ankle instability, gluteal tendinopathy, premature osteoarthritis of the hip and knee. These are not injuries caused by too much walking. They are the direct biomechanical consequence of walking on perfect, engineered, unyielding sameness that a foot built for chaos has no defense against. The practical step: walk on uneven surfaces deliberately, at least 3 times per week. Grass, gravel, a hiking trail, sand. Remove your shoes when it is safe to do so, and let your IFMs receive the proprioceptive data they were built to process. Even 15 minutes of barefoot or minimalist walking on an uneven surface begins reversing IFM atrophy and restoring the somatosensory feedback loop. Your foot needs to learn to read terrain again. Give it the terrain. 7 factory settings. 7 biological programs that your ancestors ran automatically because their environment demanded it and that your environment has systematically eliminated. Panoramic vision to shift your nervous system off high alert. Segmented sleep to use the midnight hormonal gift instead of fighting it. The deep squat to prevent your connective tissue from solidifying. Cold exposure to reactivate your metabolic furnace and protect your neurons. Active navigation to grow the brain structures most vulnerable to decline. An eating window that lets your gut run its cleaning cycle. And variable terrain to keep the proprioceptive hardware of your joints online. Not one of these costs money. Not one requires a clinic or a supplement or a device. They require only the understanding that your biology was built for a world very different from the one you currently live in, and that you have the capacity to reintroduce what that world demanded. Which one are you restoring today? Subscribe. The next video covers the ancient breathing mechanics your lungs have been waiting for.